Good morning, everybody. I welcome everyone today.
Good morning.
Good morning, ma'am.
Good morning, sir.
Yes, start. Jai sir, start.
Thank you. So good morning once again to all of you. I'm very happy that in our PhD webinar series, today we are having our 8th speaker, and we plan to continue this series for the betterment of microbiologists in India and abroad. So today we have one very special speaker with us, Dr. Joyline Mascarenhas, who is currently working as an assistant professor in the Department of Microbiology, Wilson College, Mumbai. And there is one very important detail I would like to share, that she is working as an assistant professor since 2009. Ma'am has completed her PhD in December 19, and ma'am has almost 12 peer-reviewed paper publications in national and international journals. Ma'am has published 2 books, and Dr. Joyline has received 1 UGC minor grant and 3 grants from the University of Mumbai for various projects related to bioremediation. Teaching is an interesting form of learning. So today, I would like to welcome our speaker, Dr. Joyline. Ma'am, please.
Thank you very much, Jay, sir. I will share my screen. I will also stop the video so that it doesn't interfere.
Sure. And I request everybody to please keep yourself muted unless you are speaking.
Is my screen visible, sir?
Yes, ma'am.
Okay.
Please proceed.
Good morning, everyone. Myself, Dr. Joyline Mascarenhas, and I would like to profusely take this opportunity to thank my guide, that is Professor Dr. K. Aruna, who is a retired professor right now from the Department of Microbiology, Wilson College, who has been my mentor and my guide throughout my entire PhD work. The topic of my presentation is optimization of polyhydroxyalkanate accumulated by bacterium, that is Bacillus megaterium strain JHA, from oil contaminated soil and characterization of the biopolymer. Now, since the 20th century, the age that has been called is called as a plastic age, where we are using plastic in everyday life. Like, for example, every small commodity that we are using right now is made up of plastic. The reason why plastic is being used is because it is highly flexible, adaptable, durable, strong, and we all know that it is manufactured from crude oil. Now, as per the 2017 statistics, the amount of plastic that is generated, that is around 8,300 million tons of plastic. From that, only 9% is recycled and 79% remains back in the landfills. And we know that it is one of a major problem that has resulted in global warming. And plastic has the biggest problem of being non-biodegradable in nature, which has affected the nature, flora, as well as the fauna. So right now, we as scientists need to look at what could be an alternative to plastic, which is a need for the hour. Obviously, the answer to this question is bio-based plastic, which could be obtained from animals, plants, or microorganisms. And basically, the plastic, when we call it as a bio-based plastic, it should have certain properties. That means it should be made up of 50% organic matter. 90% of it should be degraded within 6 months of duration. And the components that are released after degradation should not affect plant growth. So under bio-based plastics, we have various examples like starch, cellulose, you have PHA, polyhydroxyalkanoates, And today I will focus only on polyhydroxyalkanoates. These basically are polyesters, 3-hydroxy fatty acid monomers. We call them, or we know them more as inclusion bodies, which are present in various microorganisms. When we talk in terms of microorganisms, we know that they play a very important role in the survival, where they help to survive under stress conditions, mostly produced during nutrient-limited conditions. Now, when you analyze polyhydroxyalkanate over different organisms, you find them of different types. We have the we have the small molecules, we have medium-sized molecules, and we have large molecules. And the most important or crucial factor about this is of this biodegradable plastic is that it can be degraded to, and it can form carbon dioxide and water at the end. So the aim of this particular work was to screen and isolate a potential PHA from various samples, optimize the parameters so as to enhance PHA accumulation, then was to use an alternative carbon source so that this could reduce the cost in terms of production, extraction of the biopolymer, its characterization, and finally, biodegradation studies. So just a gist of what was done, the samples were collected and enriched. Then they were analyzed by initial screening methods. One is a lipid screening, then by Nile blue plate assay, which is a secondary screening technique. The isolates obtained by this technique positive were then used for estimation by PHA. The most probable organism or the promising isolate was then identified by biochemical studies and by 16S rRNA method. The parameters for PHA accumulation were optimized. The parameters included factors like media composition, pH, temperature. So one factor at a time approach was utilized for this study. Then a comparison was done of the optimized medium with the original medium. And to increase or to make it commercially viable, instead of using a commercial source or a laboratory medium, an alternative with the use of agro waste was considered, wherein scale-up studies were done, and the biopolymer was then characterized and studied under techniques like confocal microscopy. Characterization was done by HPTLC, FTIR, NMR, DSC, TGA, GPC. Tissue toxicity studies was done to understand if this PHA is implanted into any kind of a living system, what could be the effects? And finally, biodegradation and study of the biodegradation with the help of scanning electron microscope. So now I'm going to discuss what I have actually done during the course of my PhD, along with the results and a certain amount of discussion. Now, for the primary screening, the various isolates that were obtained were initially screened by now in case of this, 120 isolates were
Hello,
obtained. Yes.
I think your screen has stopped.
One second.
Sure.
Yes, sir. Are you able to see my screen now?
Yes, please continue. Thank you.
Yeah. Sorry for the technical glitch. Now, talking about the results, the initial part of the work, wherein 120 isolates were obtained, out of which 81 turned out to be lipid positive by the primary screening technique. These lipid positive cultures were now further studied by secondary screening technique called as a Nile blue plate assay. Now, the secondary screening technique becomes very much efficient since the Nile blue has an affinity for PHA molecules, and it becomes a concrete method by which you can detect the presence of PHA producers or PHA accumulators. So these were the different samples that were taken for study or analysis. Samples were from areas like machine oil contaminated areas, oil contaminated, effluent textiles. Now, in each of these samples, a certain percentage of organisms were able to accumulate PHA. Now, from the 13 isolates that were identified, they were subjected to PHA estimation by a medium called as E2 medium, which is basically a minimal medium using 2% glucose. And the extraction that was utilized, method was utilized was by Raute et al., a modified hypochlorite method. And the estimation, final estimation was done by the Slepley and Law method, wherein PHA in the presence of sulfuric acid got converted into a product called as crotonic acid, which could be detected with the help of a spectrophotometer at 235 nanometers. Now, the isolate that from the 34 isolate, one of the isolate that was designated as CO1 showed maximum PHA production, which was further identified as Bacillus megaterium. And this was done by biochemical studies, Vitex, as well as the 16S RNA typing. And this organism was then subjected to gene bank studies, and it was given a strain name as JHA. This organism is a gram-positive organism, and utilizing a gram-positive organism over a gram-negative was considered or is considered to be advantageous because of the simplicity that is involved in the extraction, especially at a commercial level. Now, for optimization studies, the various parameters that were used, initial parameter used was a proper method for extraction of the PHA. Now, among the methods that were used, initially the modified hypochlorite method was what was used, but better than that, the chloroform method was found to be advantageous. And this method was used for the remaining optimization studies. Now, different media, basically most of the media that were used were minimal medium with difference in the trace element components and the salts that were used. But in spite of different media used, the E2 medium continued to show maximum PHA accumulation. Parameters like pH, temperature, the pH maximum PHA production was seen at pH 8 and at temperature of around 28 degrees Celsius. And optical density was found to be, the maximum pH accumulation was found to be at OD1, and the inoculum size was around 5%. So on studying different parameters, like for example, inoculum size, one thing that was observed that the size of your inoculum affects the lag phase, which in turn does affect the PHA accumulation. Aeration speed also plays a very important role in PHA accumulation because oxygen depletion led to an effect on the TCA cycle, which also in a way helped in enabling PHA synthesis. Now, incubation period, this is a very important factor because many times when we talk about PHA, these are products that are produced during the stationary phase, and the organism was able to produce PHA after 96 hours of incubation. And increase in incubation period does affect trace element availability. It increases the metabolites in the medium, and later on there a drop was observed. So this does support the factors that are associated even in case of growth.
Hello, ma'am.
It seems there is some technical problem.
Yes, looks like that.
Yeah.
So am I audible?
Yes,
Yeah.
yes.
Okay, sorry, I think the internet got disconnected. Yeah, so can you see my screen?
Yes, please.
So the different carbon sources that were used included components like glucose, sucrose, maltose, but along with it, various types of acids and oils were also utilized. But the best carbon source turned out to be glucose. The different concentrations of glucose that were used varied from 0.5% to 10%. 2% glucose showed maximum PHA production. In the E2 medium, two components that were also present were inorganic nitrogen sources and a certain amount of organic. So among the inorganic nitrogen source that was used, microcosmic salt showed maximum PHA production. Now again, varying the concentrations of microcosmic salt, 12.5 millimolar of microcosmic salt played a very important role in maximum PHA yield. Now, one of the most important components in terms of PHA accumulation is the nitrogen limitation. And the amount of nitrogen that is present in the medium was found to be very less, and that also enables or that also enhances PHA production, which was proved when this experiment was conducted. Now, different organic sources were utilized, like yeast extract, soya peptone, and beef, and even a control was put wherein there was no organic nitrogen. But it is seen that to a certain extent, an organic nitrogen source is required because it provides the essential amino acid, vitamins, as well as minerals and trace elements. On varying the concentration of yeast extract, the amount that was utilized or gave maximum PHA accumulation was around 0.04%. On replacing the yeast extract with other inorganic nitrogen, it was studied that instead of yeast extract, KNO3 could also be, or potassium nitrate could also be used, which would show enhanced PHA accumulation similar to that as yeast extract. So henceforth onwards, the medium was now supplemented with potassium nitrate instead of yeast extract. Now, various concentrations of potassium nitrate also was utilized to check for PHA accumulation, which was found to show maximum PHA accumulation at 0.04%. Again, helping us back to the same concept that low nitrogen concentration and a good sugar content plays a very important role in PHA accumulation. Other salts that were present in the medium like K2HPO4, KH2PO4 also were studied from the point of view of PHA accumulation. And these phosphates play a role not exactly in enhancing accumulation, but they were helping in cell growth and buffering action. Now, one important parameter in terms of PHA accumulation is the carbon-nitrogen ratio. Various ratios were studied, and one that showed maximum PHA accumulation was an optimized ratio of 70:1. The initial ratio that was used was 6.6:1, that is utilizing glucose and the salts, but with increase in the carbon to the nitrogen ratio, the PHA accumulation was found to be much enhanced and better. Now, trace elements, which also plays a very important role in a minimal medium, was utilized. and the presence of trace element at a concentration of 2 milliliter per liter of the production medium showed enhanced. Increasing the amount of trace element, in fact, had a negative effect on PHA accumulation. One more trace element which is required or very essential for PHA accumulation was magnesium sulfate. But this magnesium sulfate also is required only in very small concentration. In the absence of magnesium sulfate, there was no PHA accumulation. But as you increase the concentration of magnesium sulfate, also it is affecting PHA accumulation. So to a certain extent, magnesium ions play a very important role in affecting the membrane permeability, which also affects your glucose intake. And finally, this will play a very important role in PHA biosynthesis. So this is just a comparison of the initial medium. This was the original medium that was used. And this is the optimized medium, which was later on termed as JHA medium, where the carbon and the nitrogen ratio was 70:1. The nitrogen sources now from microcosmic salt and yeast extract were changed to microcosmic salt and KNO3. And the nitrogen concentration that was utilized was in minimal amounts. So again, this is just a graph to represent the comparison of the PHA accumulation using original and optimized medium. Now, one of the problems that is associated with any kind of a commercial production is the cost that is involved. So the next part of the work that was done was to try and substitute the carbon source with some kind of an agro-industrial waste. So different agro-industrial waste were utilized, like for example, molasses, waste oil, bagasse, neera, banana peel, potato peel, papaya peel. All these were utilized in the initial screening technique. From this, molasses showed the maximum PHA yield, and hence it was used for further experimentation. Now, there were 3 types of media that was prepared using molasses. The first is where only molasses was utilized in distilled water and PHA accumulation was studied. Second was varying the concentration of molasses in the original E2 medium containing both the nitrogen sources, that is microcosmic salt, as well as KNO3, that's potassium nitrate. But a third thing that was done was varying the concentration of molasses in the E2 medium and completely eliminating the nitrogen sources. And this medium was found to be highly efficient. So when I was studying literature with related to molasses, molasses contains a large amount of sugar, trace elements, vitamins. So to a certain extent, the absence of nitrogen in a medium could still be compensated by the components that were present in the molasses, and that in turn helps in enhanced PHA accumulation. Now, in order to confirm the presence of PHA, the Nile Blue assay was done. Now, in case of this, the molasses was added into the medium and the fluorescence was observed in the presence when the Nile Blue assay was performed. Now, coming from the state of Maharashtra, where we have the largest sugar production, utilizing a source like molasses could be a very good alternative for PHA accumulation. This was the scale-up studies that was done using the fermenter in the laboratory. But yes, there is more need for standardization when we talk about a large-scale production in case of PHA accumulation. Now, further part of the work that was continued was studying these organisms in the presence of to understand the fluorescence or to understand the presence of the PHA bodies under the confocal microscope. Here, images that are seen is a presence of these bodies, inclusion bodies, in the presence of glucose and molasses wherein these cells that are able to accumulate PHA are seen as fluorescent bodies when exposed to UV light. This was a biopolymer that was obtained. This is from glucose, E2 medium containing glucose, and this was in the E2 medium containing molasses. Now, if you compare the characteristic, the biopolymer from glucose was rough, intact, translucent, whereas from molasses was smooth, glossy, brittle, and opaque. And both the biopolymers were soluble in chloroform. The biopolymers were further subjected to characterization. The first method was HPTLC, where a standard PHB molecule was utilized. Now, as you can see in the result over here, that the bands corresponded to the standard PHB, which talks about the biopolymer having certain components similar to that of a standard PHB. Obviously, since this is obtained from an organism, there are other bands that are also there, which could be because of the presence of other molecules present in the PHA. Now, on performing FTIR analysis of both the biopolymers, the functional groups observed were similar to that of a standard PHB molecule that was used, a sigma molecule, which showed CH stretching, COOH stretching, which also confirmed that the biopolymers extracted were derivatives of polyhydroxyalkanoates. Similar studies were confirmed further by NMR as well as DSC analysis. Now, DSC analysis is that is differential scanning calorimeter analysis is very important from the point of view of biopolymers, where the polymer stability and the shelf life of the polymer plays a very important role. Now, if you look at the summary over here, you can actually see that there are different peaks. Now, multiple peaks present, especially in polymer 2 that is obtained from molasses, were attributed to different impurities or presence of different large side chains. And it is considered that polymers having a melting temperature of less than 160 are considered to be potentially stable molecules. Thermogravimetric analysis that was done to study whether the components are amorphous or crystalline in nature. And the carbon source utilized could also influence the thermal degradation of these polymers. So higher the degradation temperature, higher is the thermal stability of the polymer. And this is an important factor if we are considering its elasticity and utilizing the polymer from the point of view of molding. Gel permeation chromatography of both the polymers showed the presence of two peaks. Now, the presence of the peaks will vary depending on your carbon sources and the organisms that are utilizing these carbon sources. And biopolymers with low PDI, that is polydispersity index, can be considered to be commercially viable and could be used for applications related to drug delivery. Since we are talking about PHA, one of the major applications of PHA is its utilization in medical fields such as tissue engineering and scaffold preparation and drug encapsulation. So the MTT assay was performed in order to check whether it had what concentration of the presence of this biopolymer could affect any kind of cell. So the 3T3 mouse cell line showed IC50 value as 0.3211 milligram per ml. The final part of the work dealt with the most important aspect, that was biodegradation of the biopolymer, wherein the biopolymer was subjected to degradation under aerobic conditions over a period of 30 to 90 days. And this is a SEM analysis. This is my control sample. And this is how the degradation, how it looks or appears after 30 days. Now, this was a setup that was done wherein soil was taken and it was allowed, a small section of the biopolymer was kept in the soil for degradation studies. Now, while performing the degradation studies, the polymer which undergo hydrolysis was because of the microbes acting on with the help of certain enzymes. One of the enzymes is depolymerase, which could lead to total biopolymer degradation. Now, one experiment that was done to understand this degradation was the E2 medium containing the PHA was inoculated with compost soil, and from that, different organisms were isolated, which were subjected to a test called as modified tube MPN method. And this is the film degradation test or the modified tube MPN method that was done, wherein the tube with the individual isolate containing the film was observed over a period of 30 days. So initially the film was intact. Over a period of 30 days, if you compare the control tube, which was intact, whereas the others started showing fragmentation. And while performing these biodegradation studies, especially in a pot experiment, the film showed complete degradation over a period of 60 to 90 days. So in conclusion to the entire work that was done, Bacillus megaterium strain GHA, which is a gram-positive sporulating organism, showed maximum PHA accumulation on optimization. The original medium was E2 medium. It was then replaced by the JHA medium, and the organism was able to show PHA accumulation also in the presence of molasses as a carbon source. Now, why molasses was considered was more from a commercial point of view. And on analysis of the PHA films by various techniques like HPTLC, FTIR, NMR, DSC, TGA, it was confirmed that the biopolymer obtained was a derivative of PHA, plus there was thermal stability of the biopolymer, and the molecular weight of the biopolymer was also confirmed. So, and the most important thing is the biopolymer was completely degraded within a period of 60 days. This was especially under conditions of facultative aerobic conditions. So this biopolymer could further be scaled up to perform or for further applications, wherein tissue toxicity using human cell line needs to be done if we are talking from the point of using the biopolymer for the formation of nanofibers and for drug delivery system. Biopolymer could also be an alternative to food packaging since it got degraded on its own. So even if it is getting collected in a landfill, it could be then degraded over a period of time. So these are the papers published related to the work. And now we can have questions.
Thank you very much, ma'am. Very nice presentation.
Thank you, sir.
I think in the chat box, we have our first question.
I just open the chat, sir. Yeah. What is the cost of this PHA as compared to the commercial one? Okay. That is Dr. Devyani. Dr. Devyani, I have not completely done the costing as such, but commercially available PHAs I know are extremely expensive because that is one of the factors that was being considered. Now, when the work was being done with the help of molasses also, I know that molasses is also being used as a feedstock for animals. So molasses to a certain extent also cannot be considered to be as a waste product. So I am still looking or working on the lines of utilizing other kinds of what you can say fruit and vegetable waste that could be used as a used from a very commercial point of view.
Okay, very nice piece of work. Can you hear me?
Yes, ma'am. Yeah. I can hear you.
Yeah. Thank you for answering. Thank you.
Thank you very much, ma'am. I think Halami sir is asking me, did you cast the PHB film? Yes, sir. So in due course of time with the optimization, once the chloroform was completely volatilized, once the PHB was dissolved in the chloroform and I kept it at room temperature, it directly casted or it directly made a film on its own. So that is why I was able to use that for my further part of the work. Chloroform is also a harmful solvent when discarded in the environment. Samini ma'am, if I am not wrong. Yes, I
Yes,
agree. Yeah, I agree with you that chloroform is a harmful solvent. So the thing is, ma'am, I didn't have to discard anything as such because the chloroform was completely volatilized. So the thing is that the
But ma'am, I guess you have shown you. Sorry to interrupt, but I guess
Yeah.
you have shown that for the extraction of the PHA, you have used this chloroform method.
Yes.
So that might be used in a huge amount, right?
Huge amount. So it used to be like around 10 ml because basically I needed the casting of the film, and that 10 ml of the chloroform used to be completely volatilized at the end of 24 hours. So there was
But
no
that 10 ml was
residuals.
used for the 10 ml was used for the extraction also, or just for dissolving the PHA which you have extracted? Because I've seen in your slide that it has been used as an extraction method and showed higher PHA recovery.
Yeah. So now when you talk about extraction of PHP, the PHP's affinity for chloroform is the highest. I have tried the sodium hypochlorite method also, but the efficiency or the PHA accumulation was not very high. So when the initial work is performed, the volume of chloroform that was maintained was less. Okay, so initially when I started with the work, it was with the help of sodium hypochlorite and chloroform. Then later on, I have completely replace it only with chloroform because that was helping in casting a film, which was very much important from the point of view of biopolymer extraction. So the amount of chloroform used was minimal and there was no residual chloroform because it was getting completely volatilized at the end of 24 hours.
Okay, then can you just let me know that if we think upon scaling up the production of PHA to supply the commercial market, right?
Yes.
And in that case, casting of film might not be the main aim, but the extraction of the polymer might be the aim, right? In that case, the amount of chloroform which would be used for extraction, do you think that it could be feasible extraction method for PHA at commercial level?
At a commercial level, feasibility of utilizing chloroform means it will depend on how much we are large scaling the production. But the only thing in case of this was, ma'am, that when initially I used the sodium hypochlorite, the PHA that was getting that I was extracting was not very efficient, wherein the extraction method utilized over here is only utilization of chloroform and no other chemicals were utilized. So the amount of chloroform would definitely increase. But finally, as you're saying, discarded in the environment, that is actually not happening. It's not getting discarded in the environment.
Okay, because I think if we go for commercial production, then that much amount of chloroform to be used for extraction.
Yes.
In that case, to get that chloroform amount to be volatile in the environment, again, could be, as you know, that this is chlorine.
Could be a problem. I understand what you are trying to say.
Solvent, right?
Yes. Like
yeah, it can be a problem to the atmosphere and to the environment.
I think David sir also wants
to. David sir wants to add something.
I want to give some comments on whatever the discussion going on.
Yes.
As now the doctor has asked, but most of the polymer extraction you have to use one or the other organic solvent. And when we are talking about the industrial level, after the extraction, most of the organic solvents are recovered. Here in the laboratory, we are using a small quantity, so we are evaporating. But in industrial level, either if you are using alcohol, chloroform, methanol, acetone, so they have the chemical method of the recovering after the use. So when we talk about industrial level, there is no much question about the environmental pollutions due to the use of any of this organic solvent. So this is my whatever the knowledge I am having and whatever the interaction I am having with the industry people, because we also work on the EPS productions and we are using the acetone. And then acetone we are recovering, but at laboratory level, we were able to recover only 40–50%. Then I asked some of the industry, they say we can recover more than 90%. So this is the way the method is used. Now, if there is a time, I want to ask one question.
Please.
Can I ask?
Yes, yes, sir, please.
So now, when you have replaced the glucose with a molasses,
yes, sir.
when the production was increased from 14 grams per liter to around 19 grams per
yes,
liter.
Sir. Yes.
So can you have any explanations why this production has been so much raised?
So one reason that that I had spoken earlier also was molasses that was used. Like I just to answer even one more person who has written in the chat, the molasses that was used was crude molasses. So in crude molasses, there are many other components which we actually do not know. Like it is going to contain glucose, no doubt. But other than that, there are a lot of other trace elements which was also enhancing the PHA production. So that is why when performing the experiments, I did those 3 types of experiments wherein I completely eliminated the medium, completely eliminated only the nitrogen source. So when I did those trial and error experiments, the amount of PHA was found to be more. So there are so many other elements in the molasses which was enhancing the PHA accumulation.
Okay, no. One more thing. In molasses is not only consisting of glucose.
Yes.
It is mainly the disaccharides.
Yeah.
And the other thing, when you compare, if I am not wrong, you have used 2 or 3% of glucose in a medium.
Yes, sir.
And when you have used the molasses, you have used 20% of molasses.
So the concentration, it's not 2 or 3% of glucose. As per the optimized ratio of 70:1, the
but
same
what is
was
the
used.
Highest, what is the highest glucose you have used?
Highest is 20 gram percent only, sir.
20 gram
percent.
Glucose?
Yes, yes.
Okay.
In the optimized medium.
Okay. Then, all right. Otherwise,
Yeah,
I thought it is a 2%.
You know, I understood your question. The comparison between molasses and glucose, because the amount of glucose would have been less and it becomes commercially viable as compared to molasses. But no, even with molasses, it was the amount of glucose that was utilized also was as good as 20 gram percent after the carbon nitrogen ratio was optimized.
Okay. Thank you very much. And one more thing.
Yes, sir.
There's a best way of presentations. Now, if out of 23, if some PhD students are listening, now you have gone in a correct hierarchy of the presentations, minimum the PHA productions, and step by step what you optimize. Because I have attended many PhD webinar also, I have examined many PhD thesis also. In the first table or first chapter, production will be high, then reduce, then high, then reduce. So this all the students should know. They have to frame after all the results as obtained, where to put which results, and accordingly. So congratulations for a good presentation, and it has come in sequentially. It shows what is your contribution in optimizations.
Yes, thank you so much, sir.
No,
and coming from you, Dave, sir, it's an honor because we've recently heard you at the conference when we had the international conference at Wilson College,
nice.
Which was hosted by us. So it's really commendable hearing a comment from you, sir. Thank you so much, sir.
My pleasure also.
Thank you,
yes, sir. Can I take the other questions that are there?
Sure, ma'am. I want to ask you one.
Can I ask one question? A small question.
Yes, Singh, sir.
What is the role of inoculum size in the production of your product?
So the thing is, with relation to inoculum size, what was observed that a certain amount is required, or else the lag phase that was there in terms of production was getting affected. So if that inoculum size is not maintained, then the amount of PHA accumulation after 96 hours was also getting affected. So that is why that optimization of inoculum size, which may be considered a very, what you can say, many a times people don't consider that to be a very important factor, played a very important role when it came for polymer production for or extraction for me. So that amount, especially at an optical density of 1, is where it showed maximum. And when we increased the amount of inoculum, that again was hampering the PHA accumulation.
What was the concentration you chose for your studies?
5%, sir.
5%.
Yes,
so that
sir.
was the best concentration.
That
right?
was the best inoculum.
Very good. Yeah, many people ignore this fact.
Yes,
but this
yes,
is a very,
sir.
very important fact.
Yes.
You have done very good work.
I think I should thank my guide, who was very particular about things. And she used to say that if you all are doing your PhD, you all need to consider all parameters so that you all know what you all are actually working. It may be a negative result, but the negative result also is extremely important.
Yes, sir. So what is the major application of your work?
So talking from in terms of application, as of now, its major application is in the field of drug delivery and scaffolding, wherein these are used as implants, and over a period of time, these nanocomposites get degraded on its own. The thing is, I had initially spoken about this to someone, but yes, because we talk about clinical trials and everything, it is still a initial part. That is why I was only able to do the MTT assay, and I could wind up my PhD. But yes, in future, if I do apply for a DBT grant, then I would consider this as a very good option of scaling up the production and checking in terms of nanocomposite production.
Right. Recently, I saw one very, very interesting news from Iceland. The person, he made a water bottle of agar, right?
Agar.
And the best part is it is biodegradable.
Yes.
So after the water is taken by the person, the bottle shrinks. This is a very great discovery recently.
Yes.
The agar shrinks and it is biodegradable. So even your work may lead to something like this.
Yes,
biodegradable, right?
Yes, sir. Thank you so much, sir.
Very good. Okay.
Thank you, sir. Ma'am, I just have one suggestion that can you
Yes.
Please type your email address in the chat box?
I could, sir, because there are a few more questions and they would like to.
Yeah. We will try to take as much as possible, but also.
Yeah, that's my email ID.
Joyline, ma'am, can I ask a question to you?
Yes, Nilima. Please go ahead.
Yeah. Did you try with yeast, like Aerobia lipolytica, etc.?
No, ma'am. My major concentration of the work was only on bacteria, and that's why I've not tried it with yeast as of yet.
Okay.
Someone has asked about Nirmalia extract. Let me just talk about what is a Nirmalia extract. So basically, these flowers that remain back after they are, what you can say, they are given in temples. The flower waste and the leaves waste that remain back, which is kept aside, that is basically the Nirmalia waste that was used. So what you can say, a very crude part of the work was done wherein we took this waste and autoclaved it, and the what you can say, the liquid part of it was also utilized as a part of the agro-industrial waste. So this was different types of waste. within our vicinity that we were trying to address, and even address the issue with relation to pollution and its commercial utilization for PHA.
Hello.
Yes, sir.
Yes, ma'am. I had asked the question about Nirmalya.
Yeah.
I am too working on that flower waste and PHA production from flower waste. So, have you done any kind of pre-treatment to the substrate?
Ma'am, I didn't do a lot of pre-treatment. The only method that I utilized was a simple method wherein a certain amount of it was taken and it was autoclaved. So I know that there are different kinds of pre-treatment that could have been done, but since I got the best possible result with molasses, in fact, even jackfruit seeds is another component that I am trying to explore right now, because it is very good and it is a waste which is actually being generated. I know that people consume jackfruit seeds, but it is not consumed to a very large extent. So there is a large amount of waste that is getting generated in the process. So I didn't concentrate so much on the Nirmalya, only other than the screening technique.
Okay, thank you so much, ma'am.
Welcome.
I think we can take maybe one or two more questions before we wind up.
There is a question with regards to solvent extraction. Yes, just to answer this question, solvent extraction could be a method wherein there is literature talking about utilization of the rotary vacuum evaporator for production of the biopolymer, which I actually didn't utilize. In fact, I didn't try, but yes, this can be considered when I am doing further part of the work. So even answering the question with regards to utilization of chloroform, whether the concentration could be reduced, if you're utilizing a a technique like the rotary evaporator, then yes, chloroform can obviously be what you can say, as the way sir mentioned, we can retrace it or retract it as much as possible. No, I have not done any kind of viscosity studies, sir. Vinay sir has asked, viscosity increased during accumulated PHA. I didn't find any kind of a trouble when the organism when in the production medium. There was no problem in terms of viscosity, because the PHA is accumulated within the cell. So I need to extract it on the outside. It is not coming directly into the medium. So the problem with viscosity does not occur over here. Standard molecular weight will vary depending on the organism you're utilizing, the carbon source you're utilizing. So if you compare it with the PHV, it has a molecular weight which is somewhat similar, but it is not going to be exact because the organism and the carbon sources you're utilizing are different. And if I compare it again with molasses being a crude source would also give you a variation in the molecular weight. Physical form of extraction, I utilized initially, I didn't do the physical method, but coming back to chemical extraction, one of the methods that was used was by utilizing sodium hypochlorite. But one problem that I noticed with sodium hypochlorite was PHA, when we are trying to extract it from a cell, was also getting degraded, and because of which there was no proper polymer formation was happening. So that is why I eliminated sodium hypochlorite and only relied on chloroform as a method for extraction. In vitamin B12, beet molasses is used. Beet molasses contains betaine, which requires. Yes, beet molasses also was an option, but I did not actually obtain the source. Otherwise, yes, I would also have utilized that as an alternative, as an agro-industrial waste.
Hello, sodium hypochlorite will be miscible, no? Hello.
Yes, sir.
Hello.
Yes,
sodium
Vinay, sir.
hypochlorite. I am ex-Wilsonian.
Okay, sir. Okay.
Dr. Zafar student.
Okay, sir.
In 93. I passed out in 93.
Okay, sir.
Dr. MB Zafar.
Okay.
Biochemistry.
Biochemistry. Yes.
Yeah. That time Sarang was there. Sarang sir.
Yes, Sarang sir was there at that time in microbiology department.
Yeah, if you use sodium hypochlorite, okay, then it will be miscible with your media, no?
It is
Sodium
used in the
hypochlorite.
extraction, sir. Sodium hypochlorite is used in the
The
extraction. So basically,
chloroform will be immiscible with your
so
media.
yeah, we are not utilizing it in the medium, sir. It is a downstream processing where once a cell pellet is obtained, that is being
Okay.
either subjected to sodium hypochlorite and then further with chloroform. But in due course of my work, what I observed was that the sodium hypochlorite was also affecting the PHA. So that
Okay.
is why sodium hypochlorite was eliminated and only chloroform was used for the extraction process.
Okay, my question was when we say hyaluronic acid. Okay. Hello.
Yes, sir. I can hear you.
Hyaluronic acid. So hyaluronic acid standards are there for medical application, 1 molecular weight, and cosmetic application slightly below molecular weight. Those standards are there. So my
Okay.
question to you is, what standards are there for PHA?
Standards for PHA, as in to answer your question, the standard that I used was a PHB molecule that was obtained from Sigma, a pure PHB molecule. Now, if you are talking in terms of other components, whether
No,
I
I am
have in
talking about application part. When you say
application,
drug application and nanocomposite implant, so what molecular weight is required there, and how much you can achieve?
now more than the molecular weight, what is more important is the amount that can be utilized. So that was confirmed by the MTT assay. So as per the MTT
Okay.
assay, 0.3 milligram per ml was the IC50 value that was obtained.
Okay. Okay. So you have
Microplastic
published
form.
paper on this. You have
Yes,
published
sir.
paper on this.
Yes. I have
So
published.
can I get copy of this paper?
Yes, sir. So it's available on ResearchGate. If you could just check under my name, it's available on ResearchGate.
Okay. Okay.
Yeah. Thank you, sir.
Okay.
Microplastic formation is seen during degradation. Microplastic formation is seen post extraction, and the later on part of the work is to do with degradation of the molecule. So the basic idea of this work is producing the polymer and seeing that over a period of time it is getting degraded, because that is our concern right now in the environment.
All right. I think you have
Okay,
answered.
Jay, now you can conclude.
Okay. Thank you, sir. Thank you. So thank you very much, ma'am. It was indeed very nice piece of work and very nice presentation, and also very nice brainstorming session. So thank you,
Thank you,
everybody.
sir. Thank you, Jay, sir. Thank you, Arvind, sir.
Thank you, all the participants. And if Deshmukh sir wants to add anything.
Hello.
Yes, sir.
No, because you see, I'm really thankful to Dr. Dave sir. I'm thankful to Prakash Halami, Vinay Aroskar sir. And
Thank you, sir.
hello.
Thank you, sir.
And
I think sir might have connection issues.
is to inspire young generation. And I'm very happy that Joyline also has presented very confidently. And we see that the future of nation in the arms of this young generation. Thank you all. I will thank Jay he is doing very well. So I will appreciate him. Thank you. Thank you all.
Thank you, sir. Thank you, everybody. Have a nice weekend. And
Thank you. Thank you, Jay, sir.
Thank you.
Thank you, Deshmukh, sir, for this opportunity.
Thank you.
Thank you, Jay, sir.
Thank you, everybody. See you on next Sunday.
Yeah.